Nuclear magnetic resonance is a technique that uses magnetic fields and electromagnetic frequencies to study molecular structures. For high magnetic fields, superconducting magnets are used. Lower fields can be generated by permanent or electromagnets. Samples are inserted in the center of the magnet and are positioned in the NMR probe. So the sample is located at the position of the strongest field and highest homogeneity of the magnet. The NMR probe consists of coils that are used to excite the sample and record signal responses at radio frequency. The basis of NMR is that nuclei behave like microscopic magnets in a magnetic field. They spin around the main magnetic field just like a spinning top. This motion is called precession. The frequency of the precession is proportional to magnetic field strength. Additional local field differences are introduced by changing electron densities in a molecule. A higher electron density results in more shielding and thus a lower local field and lower spinning frequency. In NMR, the frequency of the excited nuclear spins can be measured from the electric current they induce in the coil. Fourier transformation of the time domain signals results in the frequency spectrum, with a signal for each different nucleus. The different frequencies we call chemical shifts, as they depend on the chemical structure of the nuclei. In practice, spectra are measured for all hydrogen nuclei at once, which results in the ethanol spectrum with 3 different peaks at different chemical shifts. Since nuclear spins behave like magnets, they also influence their neighbors. One way this happens is through shared electrons in the chemical bonds in the molecule. We call this J-coupling. Each spin can have 2 states, along the magnetic field or against the magnetic field. As an example, we look at the 2 methylene hydrogen spins, which are chemically identical. They can have 4 combinations: 1 spin down, 1 up, both spins down, 1 up, 1 down, 2 spins up. Each combination has a different energy level and influence on the neighboring spins, except that up-down and down-up combinations have the same energy level and influence. The influence is visible as a splitting of the signal of the methyl group into 3. This is called a multiplet. Likewise, the 3 methyl hydrogen spins split the methylene response into 4 signals. In conclusion, NMR can identify molecular structures from chemical shifts and multiplets.